So basically the life form, all life forms in Earth are made up of certain biomolecules. One of such biomolecules is carbohydrate. The other three biomolecules are lipid and nucleic acid. Okay. So as we see, the carbohydrates are the major source of energy from our diet, and by definition carbohydrate actually encompasses a wide range of compounds. Okay, wide range of compounds, অনেক ধরনের compound নিয়ে তৈরি হয় carbohydrate। তার মধ্যে আমরা একটা common definition draw করার চেষ্টা করেছি। How to define all compounds that fall under the same group. This particular definition has got two parts. The first part says that carbohydrates are polyhydroxyaldehydes or ketones. The second part says what? The compounds which produce the same on hydrolysis are also called carbohydrates. That means what? There are many compounds which has no aldehyde or ketone group, but when they are hydrolyzed, they produce such compounds which act as carbohydrate, which has more than one. Polyhydroxy means more than one hydroxy group and at least one aldehyde or keto group. Correct? So if you have to have more than one hydroxy group and one aldehyde group, then what could be the minimum number of carbon atoms of that molecule? 1, 2 or 3? Minimum number of carbon atoms. If it is one carbon compound, then both the functional group cannot exist. If it is two carbon compound, then they can exist, but the poly will not be satisfied. So you have to have at least three carbon atoms on any compound to fulfill this part. Correct? So any compound which has more than one hydroxy group and one aldehyde or ketone group will be considered as carbohydrate. Flat. Or compounds which will produce the same on hydrolysis. There are certain compounds which are polymer of these carbohydrates, say disaccharides, oligosaccharides, polysaccharides. The first part of this definition will not satisfy for those compounds. For them, we have the second part. Fine? Now the basic classification. Now see, there are 4 classifications: mono, di, oligo and poly. The di has been written with slight off color. Why? Because some books they include disaccharide within oligosaccharide, telling them as 2 to 10 sugar units. Monosaccharide means which cannot be hydrolyzed further to simpler sugar forms. They are called monosaccharides. Disaccharide means which produces 2 monosaccharide units on hydrolysis. Oligosaccharide which produces more than 2 monosaccharide units on hydrolysis. Polysaccharide means they are polymer of number of at least more than 10 number of monosaccharide units. Now we come to classification of monosaccharides. We can classify monosaccharides based on their functional groups presence. If the functional group is aldehyde, we call them aldoses. If the functional group is keto, then we call it ketoses, right? Examples: glucose, galactose, mannose, maltose, these are all aldoses. Ketose: fructose, iodose, xylulose, ribulose, pseudoheptulose, etc. This is a basic structure. Can anyone tell me which forms it is drawn in? What do you call this form of drawing structures of molecules? বলো Fisher straight line Fisher form, right? So the Fisher form যদি আমরা দেখি glucose and fructose, what is the difference? See the structure. They are different in two positions, C1 and C3. Check out. In C1, that is functional group is aldehyde, and C2 in ketose, fructose is keto group, the functional group, and C3 is different, having OH on, sorry, C1 and C2, and rest all are same. C2, 3, 4, 5, 6, all are same. Only the difference C1 and C2 because of the functional group. Number of carbon atoms. So minimum number of carbon atoms should be 3. So it should be trioses, then tetroses, then pentoses, hexoses, heptoses, octoses, nonoses, and docoses. Then stop. Right? See, some examples have been given. Huh? Now we come to disaccharide. Easy. Just make a chart, tree like this. Disaccharide can be classified based on their reactions. Some of the disaccharides are reducing and some of them are non-reducing. Reducing means they can reduce cupric sulfate. Cupric sulfate or copper sulfate. How? There is a reagent called Benedict's reagent. When we react with that, they reduce CuSO4 to Cu2O, plus oxide, red color precipitate. And this reduction takes place if they have free carbonyl group, means aldehyde or keto group. So if there are certain disaccharides which has no free reducing ends, they cannot reduce Benedict reagent, Fehling reagent, etc. Examples: reducing is easy to remember. Non-reducing, you see, sucrose and trehalose. What is sucrose? Cane sugar or table sugar. Trehalose, in our blood, what is the function of glucose? দাঁড়াও তুমি। In our blood, what is the function of glucose? Provide energy or nutrition, correct? The same function trehalose has in the blood of insects. Correct? So, trehalose is actually a dimer of glucose. This is number 1 carbon and this is number 1 carbon. These two carbon participate in formation of this linkage. This linkage is called glycosidic bond. So what type of bond is present in trehalose? It can be written like this. Okay, like this also you can write 1 arrow 1 1 comma 1 means 1 1 glycosidic linkage. Similarly, sucrose has this is C2 and this is C1. So the linkage is beta 1 2. You can write in any way. Why? Because it is so well known, no one can do mistake. Everyone knows. It is between second carbon of the fructose and first carbon of glucose. So that is why in these two structures there is no free end of aldehyde or keto group. So they cannot participate in reducing reaction. Easy. For trehalose, there is an enzyme in insect's body called trehalase. Which cleaves this glycosidic bond, and glucose is released and can be utilized by the insect cells. Yeah. Do we have this enzyme? Who has it? Insects. Do we have enzyme which can cleave this? No. Then why? How come you digest sucrose, table sugar? Do you like sugar? Can you eat sugar and digest? Then we have, for maltose the linkage is alpha 1,4. For lactose the linkage is beta 1,4. For isomaltose the linkage is alpha 1,6. Right? Maltose ta alpha 1,4, lactose ta beta 1,4 আর isomaltose alpha 1,6. খালি ল্যাকটোজের ক্ষেত্রে কি আছে, দুটোই গ্লুকোজ নয়, একটা গ্লুকোজ আর একটা গ্যালাক্টোজ, বিটা গ্যালাক্টোপাইরোনোসিল 1,4 alpha D glucopyranoside. এগুলো নাম ওই বড় বড় নাম হয়, তাই না? বলো তাহলে now we know about some disaccharides. Now we should know that enzymes also, like this trehalose, trehalase, sucrose, digesting enzyme sucrase. For maltose, maltase. For isomaltose, isomaltase. For lactose, lactase. Now, unlike these two, maltose and isomaltose, lactase enzyme deficiency causes havoc. It's called lactose intolerance. It's called lactose intolerance. Now, for us, no problem. We are grown up. But for newborn, they cannot digest mother's milk. They cannot digest lactose because of deficiency of lactase enzyme. The condition is very, very dangerous for newborns because we can refrain from eating milk, drinking milk. Newborns cannot. So they suffer, they present with some symptoms. What are those? Now, demographically মানে বলো demographically আমি কি বললাম, ফার্স্ট ইয়ার হচ্ছে অ্যালফাবেট, হোয়াট ইজ হোয়াট জানো, না জানলে আমার জিজ্ঞেস করো, আমি আছি তো। এখানে কোনটাকে কি বলে এইটুকুন জানবে ফার্স্ট ইয়ার, আর কিচ্ছু না। ডেমোগ্রাফিক্যালি মানে হ্যাঁ বলছে এরিয়া ওয়াইজ, কারেক্ট, রিজিয়ন। তো ডেমোগ্রাফিক্যালি আমাদের যদি ইন্ডিয়া দেখো, এটা ইন্ডিয়া মনে করো, কাশ্মীর, গুজরাট, রাজস্থান রাগ করো না, হ্যাঁ? If you draw a line like this, then the people above the line can digest milk naturally. People below the line cannot digest milk. That's why we use, what is curd? Curd is something, in a phrase they say, when milk sleep night, morning tight. It's called curd. রাত্রিবেলা দুধ যদি ঘুমিয়ে পড়ে সকালবেলা উঠে টাইট হয়ে যায়, তাকে বলে কার্ড। কার্ডে কি থাকে, ল্যাকটোজটা ডাইজেস্টেড হয়ে যায় বাই ব্যাকটেরিয়া, ব্যাকটেরিয়া ল্যাকটোজটা খেয়ে ফেলে, প্রোটিনটাকে খেয়ে ফেলে, খেয়ে প্রোটিনটাকে ভেঙে দেয়, আরো সহজপাচ্য প্রোটিন তৈরি করে। more easily digestible protein products are formed from milk proteins by bacteria. So curd is easier to digest without any sugar. No milk sugar is present. Clear? That's why we can digest curd, but we cannot digest milk. Now, what happens when milk reaches gut of a person who is suffering from lactose intolerance? Usually, people from this area, they develop lactose intolerance during adulthood. Usually, they develop lactose intolerance during adulthood. But some unfortunate patients, they are born with this enzyme deficiency. Their problem is like this. Lactose, being osmotically active molecule, when reaches the gut, particularly large intestine, it draws water inside. Right? Why? Because lactose is sugar, it's osmotically active molecule. It will draw water inside. What will happen? The large intestine will be full of water. Now, we have two different types of sphincter. One sphincter which will disallow liquid, another sphincter which will disallow solid. So, urinary sphincter is such which disallow passage of liquid, but gut or anal sphincter which will disallow passage of solid. But in this case, it is not solid. So what will happen? Frequent passage of stool. What is that? One word acronym is diarrhea. Lactose remain undigested, so what will happen? This will be acted upon by the bacteria will feed on lactose, will produce sulfur dioxide and carbon dioxide. So what will happen? The stomach, abdomen will be like mine, and it will make sound, not flat sound, sharp sound. That means full of, that is called पेट फापा. Next, release of gas. Right? So these are the symptoms and why they happen like that. Do you have a treatment? Yes. Avoid lactose containing substances. Avoid mother's milk, breast milk. What will give? In market, the formula milk is available, lactose-free formula milk. You can prescribe that. এটা অস্বস্তি বটে, সিমটম জিনিসও বটেই। হ্যাঁ, সিমটম হবে, তাই না? আর সিমটমটা কার হবে? আমার তো হবে না তো। নিউ বর্ন, 1-2 দিন বয়স, মায়ের দুধ খাবে, পেট ফুলে ফেপে মরে না সাধারণত। বুঝতে পারে, মা বুঝতে পারলে ভালো, না বুঝতে পারলে কাঁদছে কেন, আরো খাওয়াবে, আবার খাওয়াবে। লাইফ থ্রেটনিং নয়। আচ্ছা, অলিগোস্যাকারাইড। দেখো, অলিগোস্যাকারাইড এর ডিটেলসে আমি এখন বলবো না। অলিগোস্যাকারাইডটা একটু জটিল, মোর কমপ্লিকেটেড, যাবো না। ও লিঙ্ক ডাইস্যাকারাইড আর এন লিঙ্ক, সরি, এন লিঙ্ক অলিগোস্যাকারাইড, এই দুটো হয়। ও আর এন। কেন ও আর এন? এই জন্য ও আর এন। ও মানে হাইড্রক্সিল গ্রুপ অফ সেরিন অ্যান্ড থ্রিওনিন। কি কি আছে? হাইড্রক্সিল গ্রুপ কন্টেনিং অ্যামিনো অ্যাসিড 3টে আছে, এখানে দুটো লেখা আছে, সেরিন, থ্রিওনিন আর টাইরোসিন। এই 3টে অ্যামিনো অ্যাসিডে OH group থাকে। These 3 amino acids contain hydroxyl group. When an oligosaccharide chain is attached to this oxygen, it is called O-linked. And there are other amino acids in the proteins like asparagine, glutamine. They have extra amino group. In this case, the growing chain is attached to the nitrogen of amino group, hence it's called N-linked oligosaccharide. O-linked and N-linked. What are the function? They function more like cell surface receptors, like our blood group antigen. Jolka, drink water. Jolka. You want a sign of dehydration. So, O-linked এবং N-linked oligosaccharide, they are part of cell surface receptor. যেমন blood group antigen. কি কি আছে antigen? A, B, O, AB, তাই না? এই সবগুলোর মধ্যে এই oligosaccharide পাওয়া যায়। ঠিক আছে? আপাতত এইটুকু মনে রাখো। For the time being, this much is sufficient. এখানে একটা স্ট্রাকচার দেখানো আছে, যেমন এই অ্যাসপারাজিনের সঙ্গে দেখো NAG মানে FUC মানে ফুকোজ, NAG মানে গ্লুকোজামিন নাক আছে, হ্যাঁ ন্যাম, কোথায় গেল? ম্যানোজ, দুটো ম্যানোজ, আবার নাক, আবার গ্যাল মানে গ্যালাক্টোজ, NN N-acetyl neuraminate. Where sometimes in question they ask which of the following is a nonose, means 9 carbon containing carbohydrate. They will give many names including this one. So remember, this is an example of nonose, 9 carbon containing carbohydrate. Okay? Sometimes they will ask like this. MCQ will. Polysaccharide. Two types, homopolysaccharides, heteropolysaccharides. Homopolysaccharides কাকে বলে? Where all the sugar units are identical. যেমন কমলা রঙের গাঁদার মালা, orange color marigold chain, all flowers are of same color. Another garland is available in the market, one orange and one yellow. So two different, so they are like polysaccharides. Examples: starch, glycogen, these are cellulose, these are homopolysaccharides. Heteropolysaccharides: heparin, heparan sulfate, darmatan sulfate, chondroitin sulfate, right? All these. Don't worry, we have to read everything in detail. Fine. Just remember the examples for the time being. Now, a short question about isomerism. Among the glucose, the isomerism is slightly different compared to those in amino acids. Whatever isomerism things I will explain, do not mix up with amino acid thing. Slightly different. Isomerism, this is what you have studied in +2. If you can remember the Bell and Bell organic chemistry book, isomerism first page, this type of diagram was there. I don't know, still you read Bell and Bell organic chemistry book? Anyone? NCERT, oh something like that. Anyway, so in our time we used to read Bell and Bell organic chemistry book. So this was there. So you have idea about these isomerisms, but we will not study all these isomerisms here. Few things you must remember. What is that geometrical isomerism? See the arrangement of bulkier groups on either side of the double bond. It's called trans. Same side of double bond. It's called cis. What is the difference between optical isomerism and geometric isomerism? Very good. It has nothing to do with geometry. It is completely different issue. Correct? This is India and Bangladesh or Pakistan, and that is Israel and Palestine. Completely different. Very good. Optical isomerism is something which is completely different. This is something like how you look at it, look at the molecule. Huh? These 5 points we will remember under the heading of isomerism in carbohydrate. First one is capital D and L isomerism. Second one is small d or positive, small l or negative isomerism. Third one is epimerism, anomerism and pyranose and furanose isomerism. That means I have to finish this chalk and then go, huh? Good. Now, what is this? You have any idea? This is D and L isomerism. There is a mirror. Which one is shifted? Right, left? Which one? Which carbon? How do you number them? This is second carbon or fifth carbon? Huh? How many of you think this is fifth carbon? Fifth, number 5. And how many of you think this is second carbon? Fifth carbon. ও কটা হয়ে গেল 4 আহা বেশি হয়ে গেছে বেশি হয়ে গেছে দাদা 1 2 3 আরে এত বেশি কাটলাম কেন দাদা বেশি হয়ে গেছে 2 3 4 Now this is 1, 2, 3, 4, 5 and 6. The fifth OH is on right hand side. Then we call it as D isoform. The L is just opposite. Only the fifth or penultimate. Means last but one. If it was pentose sugar, then it would be C4 If it is hepto sugar, then it will be C6. Correct? Similarly. So penultimate carbon, you must look for penultimate carbon. If the OH is on right hand side, we call it D isoform, capital D. And if left hand side, we call it L isoform. That's all. No big issue. It was made by Fischer based on 2D representation of this molecule. Correct? Next comes small d, which stands for dextrorotatory, and small l, which stands for levorotatory. Now, glucose can be of either of these state, D or L, right? But this small d and small l is an inbuilt property. It cannot be changed. Correct? That means if they rotate the plane polarized light clockwise, they are called D or correct. If they rotate the plane polarized light anti-clockwise, they are called L. Glucose sorry, বুড়ো হয়ে গেছি, বলি এক লিখি আরেক। So glucose is dextrorotatory, fructose is levorotatory. Glucose এর কত? এটা anomers, এগুলি পড়ালাম না, দরকার আছে? দরকার নেই তো, আমার না ওখানে এক দরকার আছে, আমার দেখিস নোকে দেয়, ওই আবার আমার তাকিয়ে আছে। When an aldehyde group আর পাচ্ছিস না, আর পাচ্ছিস না তো, জল খা, আবার পারবি। When you run out of fuel, you put fuel and then again run. Drink water, start again. Drink. খা, এই তো সবে কলেজে এলি, প্রথম ক্লাস করছিস, কি স্কুল? সকালবেলা মা টিফিন দিয়ে দিল, গিয়ে খেয়ে নিলাম, বাড়ি চলে আসলাম ড্যাং ড্যাং করতে করতে। হ্যাঁ, সেই দিন গেছে, আর নেই। অ্যালডিহাইড অ্যান্ড কি? হ্যাঁ হ্যাঁ হ্যাঁ, আছে আছে, পড়াবো পড়াবো, চেঞ্জ করে দেবো। 112 আর প্লাস 12, দুটো মাঝামাঝি হয় 18, 112 আর 18, 18.8 এটা 19 ধরে, 111.2 যেটাকে 12 ধরা হয়। আমি জানি ওটা, পড়িয়ে দেবো, নিশ্চিতে থাক, তো you will never run out of material. Alcohol or aldehyde, when they combine they form acetaldehyde. কি লেখা আছে আমাদের ওখানে দেখ, acetal. Why it is hemiacetal? When the alcohol and aldehyde within the same molecule, then we call it hemiacetal. Similarly, when keto group and aldehyde, alcohol combine, they form hemiketal if those groups are in the same. That means what? Aldehyde, alcohol forms a bond. Correct? Now the new bond, what is formed due to that, C1 character has changed. It has now two different hands. In one hand it has got hydroxyl group, another hand it has got hydrogen group. If the hydroxyl group is on right side, we call it alpha. If the hydroxyl group is on left side, we call it beta. Because in Bangla, beta stands for left. And till few years back, left was in government. Till few years back. So, when we draw the diagram like this, right and left, alpha, beta, we understand. What happens when we draw the diagram like this? Considering Bengal before 2011, when left was in government, left upward is called alpha. Sorry, beta. Left, beta, upward. And the opposite is alpha. Then this OH is fixed D, so it will come D. Right? Then these structures are similar to this structure. Pyran compound. So there, pyranoside. Similarly here, pyranoside. This is alpha D pyranoside. they resemble this compound, which is called furan. Correct? বোঝা যাচ্ছে কিছু কিছু? একটু বোঝা যাচ্ছে তো? সেটা পড়া আছে তো দেখবি। খালি কতটুকু পড়তে হবে, জাস্ট আউটলাইনটা জেনে রাখো। So when they combine, they form like this, alpha and beta. These alpha and beta are termed as anomers. So anomers are epimers. Anomers are epimers at C1 in case of aldoses and at C2 in case of ketoses. Anomers are epimers at C1 in case of aldoses, at C2 in case of ketoses. The new chiral center is formed due to formation of hemiacetal or hemiketal ring. This new chiral center, C-H-I-R-A-L, chiral center is formed due to formation of hemiacetal or hemiketal ring structure. Hello? Now we come at epimers. এই দুটো লাইন মুখস্থ করো। Epimers are isomers differing in one or single carbon atom, in only one carbon atom. They differ in the structure, in orientation by only one, any one. That's why I said anomers are epimers at C1 in case of aldoses and at C2 in case of ketoses. So apart from these, there are many other sugars without anomerism, they might show epimerism. So for example, glucose and mannose, they differ in C2. Only C2 is different in their structure. Huh? Glucose and galactose, they differ in C4 in structure. That's all. Next is ring structures. If we compare with them with 6-membered pyran form, we call it pyranoside. If we compare with them 5-membered furan form, then furanocide. Usually glucose preferred to exist in pyranoside form. Usually glucose preferred to exist in pyranose form, and fructose prefer to exist in furanose form. Fine? এগুলো মনে রাখবে, এগুলি পরে আসছি। আগে যারা খুশি হয়েছে তাদেরকে বলে দিই, এই যে সিরিজটা আঁকা হয়েছে, this series is very very important. If you can remember how to draw this series, then you can remember the names, and if you can remember the names, you can draw the structure. First, let me tell you, this is an aldose sugar series. Starting from ছবি তুলিস নি বাবু, ছবি সব বইতে ছবিগুলো দেওয়া আছে। 100 বছর ধরে আমরা পড়ে আসছি। মোবাইলে ছবি তুলে শুধু ডেটা নষ্ট করিস না, ডেটা নাও কি বলে, স্পেস নষ্ট করিস না, একদম নষ্ট করিস না, বরং যেটা বলছিস শোনার চেষ্টা কর, শুনে বোঝার চেষ্টা কর, তাই না? এটাকে বলা হয় ডি সিরিজ অফ aldose sugar, starting from glyceraldehyde, D-glyceraldehyde, starting from D-glyceraldehyde. D-glyceraldehyde structure দেখো, CHO, CHOH, OH ডান দিকে, CH2OH, 3 carbon compound. Next, it is increasing by 1 carbon in every vertical step, increasing by 1 number of carbon, by number. By convention, convention means no rule. we all sat together and decided, yes, this is to follow. By convention, when we write the structure on left hand side, we add the OH group on newly added carbon atom on right hand side. By convention, when we draw the structure on left hand side, we add the OH group on newly added carbon atom on right hand side. In case of aldoses, the newly added carbon atom is C2. C2 হচ্ছে নতুন কার্বন। তো C2 তে OH টা কোথায় আছে? ডান দিকে। C2 তে OH টা কোথায় আছে? বাঁ দিকে। বাঁ দিকে আছে ডান দিকে OH, ডান দিকে গেলে বাঁ দিকে OH। নাম কি হবে? পরে আসছি। Forget about names. Names you will remember. So this is rest all will remain same. This part is same. Why this is same? Because this is D series. Had it been L series, এই OH টা বাঁ দিকে হতো। L series হলে OH টা বাঁ দিকে হতো, left hand side. D series মানে এই দুটো fixed. নতুন কার্বন, new carbon introduced here will have OH on right hand side. New carbon introduced here will have OH on left hand side. So now there are erythros and trios. Next step, vertical step, one more carbon is added. Right? So each one will have two isoforms. Each one will have two isoforms. Now this one and this one, what is the relationship between them? They are epimers. These two 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 are epimers. Are they? No, not always. See, not always. In this case, glucose and mannose, glucose and galactose are epimers, but others are not. Right? এইখানে হবে epimer. Right. So erythros, trios, then ribose, arabinose, xylose, lysose, then allose, altrose, glucose, mannose, gulose, iodose, galactose are talose. You have some mnemonics to remember the names. If you want, I can give you. ET. Two more minutes. কি লিখলাম? ET একটা সিনেমা ছিল, there was a movie called ET, erythros and trios, ring all xylophone loudly, ribose, arabinose, xylose, lysose, allose, altrose, glucose, mannose, gulose, iodose, galactose, gal, and talose. Right? Very easy to remember. If you remember like this, you can remember the series. Fine? That's all for today.